This is a lecture on Chapter 13, dealing with viruses, viroids, and prions. And my name is Professor Louis Nell of the Department of Microbiology and Plant Pathology. So, viruses are some of the most important pathogens in today's world. They cause multiple very important diseases. They are very small. They are much smaller than bacteria, so they cannot be seen with a light microscope, and they pass through filters that would keep back bacteria. And they cannot be grown outside of their hosts or outside of living cells. And that is why we refer to them as obligatory intracellular parasites. They contain DNA or RNA, not both. They have no ribosomes, and they don't produce energy. So they don't have an ATP generating mechanism. They contain a protein coat that surrounds the nucleic acid, whether that's DNA or RNA, and then they multiply inside living cells by using the synthetic machinery of the cells. So they would take over the host cell and redirect the machinery of the cell to in fact replicate themselves. Table 13.1 shows a very nice comparison between bacteria and viruses. So again, viruses are always intracellular parasites, not so for typical bacteria. Typical bacteria and chlamydia have a plasma membrane and divide by binary fission. Not so for viruses. And viruses, yes, pass through bacteriological filters because they are so small, and they do not possess both DNA and RNA, do not produce energy, not ribosomes, and very important, they are not sensitive to antibiotics. So bacteria and rickettsias and chlamydias are sensitive to antibiotics, but not viruses. On the other hand, viruses are sensitive to interferon, which are small glycoprotein molecules that are messenger molecules that can be passed between cells to warn cells of a pending invasion or viral infection. Some viruses are enclosed by an envelope. Um, some viruses have spikes, and most viruses infect only specific types of cells in a specific host. And so, for example, hepatitis viruses uh, have an affinity or a tropism for liver cells, and they would then specifically infect the liver cells of their host of preference. In this host range, the number or the different species that can be infected by a virus, that host range is determined by very specific host attachment sites, which the virus needs to recognize to be able to infect, and also multiple other cellular factors. In this figure, we see a demonstration of different sizes of bacteria, uh, eukaryotic cell, and of viruses. You can see viruses are always very much smaller than a bacterium. In this case, E. coli, which is a typical bacterium. So always very, very much smaller, and of course very, very much smaller than the eukaryotic cell. In this case, a human red blood cell. So some of the smallest viruses infect bacteria, and viruses that infect bacteria we call bacteriophages. And you can see in this case that phage F2 is 24 nanometers in diameter. That's very small. Poliovirus is a human virus, also very small, 30 nanometers in diameter. Rhinovirus uh, falls in the same family as poliovirus, also infect humans. Rhinovirus causes common cold, um, and also small, 30 nanometers in diameter, and so on. So some of the larger viruses are vaccinia um, virus. These are the viruses that cause smallpox. It's 300 by 200 by 100 nanometers. And an interesting Ebola virus, which was very much in the news in recent years, a very long virus particle, 970 nanometers is an average length. Ebola virus particle. So these particles, a fully developed infectious particle, are called virions. So if we look at the virion structure, then it contains nucleic acid, which is DNA or RNA, but then it can also be single-stranded or double-stranded or linear or circular. So all of those possibilities exist in the range of known viruses. Then the nucleic acid is packaged inside a capsid, which is made up of protein, and the different protein subunits that make up the capsid are called capsimers. Some viruses are then also enveloped, and some have X. It's a figure showing a polyhedral virus on the left. So you can see the individual capsimers making up the capsid with the nucleic acid inside. So this is called a polyhedron, which is a specific shape, um, the giving it sort of a round appearance, but you can see it has flat surfaces and corners. And on the right, you can see an electron micrograph of such a polyhedral virus, in this case must adenovirus. And you can see the adenovirus also has spikes that can protrude from the capsid. And it can be seen very nicely in this graph. And this is another type of electron micrograph, again showing must adenovirus, and you can clearly see the individual capsimers that are the protein subunits of the capsid. In this case, we have an enveloped helical virus. So as opposed to the icosahedral particle, the capsid in this case is a long structure or a helical structure. And you can see the capsimers on the outside and then the nucleic acid within. And on the right is an electron micrograph of such a enveloped helical virus, in this case influenza virus, causing flu. And this is a good example of an enveloped virus, but where the uh, capsid is helical and packaged within this envelope. Herpes virus is an example of an icosahedral particle. You can see the capsimers in this roundish structure, so icosahedral, but this is then also um, surrounded by an envelope, as you can see in this picture. A helical virus without membrane is on the left. This is in fact a tobacco mosaic virus. You can see the individual capsimers and you can see the nucleic acid then packaged within. So tobacco mosaic virus infect obviously plants, tobacco, among others. And on the right, we have this animal virus also infecting humans, Ebola. Has been in the news quite a bit, but this is an example of a helical capsid in an animal virus. Some viruses have a complex morphology. This is, for example, seen in the T. even bacteriophages, so these viruses that infect bacteria. You can see tail fibers, a sheath, and then the capsid on top of that with the nucleic acid then within it. And on the right's a very nice picture showing how these bacteriophages recognizes and attach to very specific sites on the host cell. So they attach to that, and from there the infection can then commence. Now, not all bacteriophages look like this, but some do, and it makes a nice picture. It's kind of a moon landing vehicle. At the bottom of this picture figure, we have autopox virus. So pox viruses, um, they cause smallpox, and it's a complex, large virus particle. I refer back to the figure that 13.1 showing the various different sizes of viruses. So the pox virus is in there as a large virus, but it's a complex morphology and a complex structure. In terms of the taxonomy of viruses, family names always end in viridae, genus names end in virus, and the specific taxonomy is regularly revised and updated and determined by the International Committee on Taxonomy of Viruses, or the ICTV. So viral species are defined as a group of viruses sharing the same genetic information, same structure, and the same ecological niche. Common names are used for species, and then subspecies are designated by a number. And here are some examples. On the left, we have herpes, so this is the family, Herpes viridae. The genus, Herpes virus. And then the species, common name, human herpes virus. And the subspecies is then numbered, so human herpes virus type 1, type 2, and type 3. On the right, another example, family Retroviridae, genus Lentivirus, and the common name of species, human immunodeficiency virus. And then the subspecies, human immunodeficiency virus type 1 and human immunodeficiency virus, or HIV, type. So viruses must be grown in living cells. So it can be grown in bacteria for bacteriophages, and bacteriophages would form plaques on a lawn of bacteria, and we'll see just now what that looks like. And animal viruses must then be grown in living animals, or can be grown in embryonated eggs, or in cell cultures. So cell cultures these days are much preferred because of the advances, technological advances in growing cells in culture, and then minimizing the need to use living animals to grow virus, and also embryonated eggs, which can be a complex and a very expensive process. And we do get what is called continuous cell cultures these days, which are cells that grow indefinitely, and and that makes cell culture so much more useful for the growth of virus. So here we can see bacteriophages that infect a lawn of bacteria on this agar plate or petri dish. And the sites where the bacteriophage or the bacterial virus infected and killed the bacteria, these can be seen as clear zones. And these clear zones are called plaques. So these are very um, uh, defined infection. This is how this is achieved, typically by doing a serial dilution. If you want to, this will also allow you to determine the titer of the virus, or count them. So you can see the serial dilutions up to a certain point, and the serially diluted virus are then incubated with bacteria on the agar plate, and where they then will form plaques, which you will be able to count at the correct dilution and work it back to determine the number of virus particles in the original. So for animal viruses, we can use the live animal, which is not the preferred mechanism, but also embryonated egg. And in this figure, you can see such an embryonated egg, and you can see the inoculation of such an egg. The viruses will then grow on the membrane associated with the inoculation site. So there are different inoculation sites possible, and the location where you want to inoculate is determined by the affinity for the specific virus that you want to grow in this egg. As I said, cell culture is actually preferred. We talk about continuous cell cultures. Let me show you how that works. So here you have the tissue that is treated with enzymes to separate the cells. The cells are then suspended in a culture medium, which contains all of the nutrients necessary to sustain and support the growth of these cells in culture. And they will then grow and attach to the surface of the glass or the plastic container that you use. And normal cells will grow into a monolayer, so that is a single layer of cells, and then they would stop to grow. But transformed cells also, these are basically cancerous cells, also known as continuous cell cultures. They do not grow in a monolayer, they don't stop, they continue to grow and can then form these clumps of cells. And as I say, it is often useful to use continuous cell lines for the growth of virus. So viruses can be identified by the effects that they have on cells in cell culture. So often this is the case. And these effects, because the viruses infect the cells in the cell culture, and then they change these cells, often kill them, and these effects that you then see in the cell shape or status, these are called cytopathic effects. Other common tests used for viral identification are serological tests. So the serological tests are dependent on the interaction between antibodies and antigens. And that is a very specific interaction. So, for example, the antibodies against a specific virus can be detected in a patient should you want to do a diagnosis. You can also use antibodies to identify virus in various different types of tests. Here are three: a neutralization test, a viral hemagglutination test, uh, and a Western blot test. Viruses can obviously also be identified by the sequence, the genetic sequence, so the nucleic acids. And there are several methods. PCR is most common and is very often used these days as a quick mechanism to very accurately identify a specific virus. So these are cytopathic effects. The cells are distorted or killed, and this can be seen under a microscope. Now, for serological tests, in this case, we talk about an ELISA. So ELISA stands for Enzyme Linked Immunosorbent Assay. And you can see that if an enzyme is added to the reaction mixture, then it will produce a product that causes a visible color change if the antibody, specific labeled antibody, binds to that specific virus. And so if the substrate is then added, you can very clearly see um, the change in color on such an ELISA plate. So let show you what here is an example of a plate, and you can see the yellow area. So these are dilutions in various samples, and you can see those wells that have turned yellow indicates a positive reaction um, for a specific virus or for a specific antibody. The Western blot is a little bit more complicated. Um, the and I'm not going to go into that in any detail in this lecture. Just to know that the Western blot is a mechanism that can be used to determine the specific viruses or antibodies in a infection. This is what the result in the end looks like. In this case, it is a test for HIV in a patient. So 1 is the positive control, 2 is a negative control, which is negative. A is a patient which is negative. B is a patient which is inconclusive, so other tests would need to be done. And C is a patient that are clearly positive for HIV infection. So the polymerase chain reaction is a mechanism and a method that you are familiar with. It's a method used to exponentially amplify very specific genetic material. And so you can see in this diagram how it is used. You need specific primers, you need building blocks for the reaction, so those are the nucleotides or the deoxynucleotides, the NTPs, and then the enzyme that causes the reaction, which is a very specific heat resistant DNA polymerase. That is the basic mechanism of exponentially amplifying nucleic acid to a level where you have several millionfold of copies that can be visualized, sequenced, or um, used in any other way to provide specific information on the. For bacteriophages, there are two types of replication cycles. The first is called the lytic cycle, and the second is called the lysogenic cycle. So let's look at the lytic cycle. In this cycle, so for example, the T. even bacteriophages go through this replication cycle. So the viruses, the bacteriophages, they attach to the cell, to the bacterial cell that they're going to infect. So they attach through the tail fibers to the cell, and then they would penetrate. So the phage lysozyme, that's an enzyme, opens the cell wall, the tail sheath contracts, and then forces the tail core and the DNA into the cell. Once the DNA is in the cell, we get biosynthesis, and that is then the production of phage DNA and proteins to make new virus particles. The fourth phase is maturations or maturation, and this is where the phage particles are assembled to release the newly produced virus particles. Again, is the enzyme, the phage lysozyme, that breaks the cell wall, and the new virus particles can then be released, and then they're ready to infect new bacterial cells. So I remind you just again of this figure 13.5. On the top right, you see how these phages go and attach to the host cell in that manner, and then manages to introduce the DNA into the cell and to progress the virus replication. In figure 13.11, a diagrammatic explanation of the lytic cycle of the even bacteriophage is shown. So there you've got the 5 stages: the attachment, the penetration, where the phage injects its DNA, then biosynthesis, where the phage redirects the cellular machinery of the bacterial cell to synthesize viral components. And these viral components are then assembled in what we call the maturation phase, and then finally release, where the host cell lyses, burst open, and the new virions. If the cells burst open, they die, and this is where you see the clear zones or plaques that we've discussed before. Figure 13.10 shows what we call a viral one-step growth curve. So no new infective virions are found in a culture until after biosynthesis and maturation have taken place. So just to compare the two cycles, the lytic cycle and the lysogenic cycle, in the lytic cycle, the phage, as we just discussed, causes lysis and then the death of the host cell. In the lysogenic cycle, in principle, the what we call prophage DNA, that's the DNA of the phage, but we call it in this case a prophage because it's incorporated, built into the host DNA. Then we get what is called phage conversion and specialized transduction, and we'll talk about. So figure 13.12 shows the two cycles. So in orange we have the lytic cycle, and in green the lysogenic cycle. So let's just look at the orange cycle, which is the lytic cycle. So the phage attaches to the host, introduces its DNA, then in step 2 the phage DNA becomes circular and it enters the lytic cycle. New phage DNA and proteins are synthesized and assembled into virions, as you can see in 3a. In 4a, the cell then lyses, bursts open, and it releases the new phage virions. But after step 2, if the virus determines to go into the lysogenic cycle, so then you get where the green arrow which says or and moves to 3b, where the phage DNA then integrates, is built into the bacterial chromosome by recombination. And this then becomes, when it's integrated like that, we call that DNA the prophage. In 4b, you can then see the lysogenic bacterium reproduces normally by cell division. You can get many cell divisions, and then that's in 4b, and then in 5, occasionally the prophage may excise, it will be cut out from the bacterial chromosome by another recombination event, and this then initiates a lytic cycle. So then you go back to 2 and completes the orange cycle. Now let us consider the multiplication of animal viruses. So like bacteriophages, viruses of bacteria, animal viruses also have attachment sites that attach to complementary receptor sites on the host cell surface. So the viruses attach, and then they would penetrate by a process that we call endocytosis or fusion. We then get uncoating of the uh, of the virus particle, so removal of the capsid, and this can be done by viral enzymes or by enzymes supplied by the host. Next we get biosynthesis, that is the production of nucleic acid and proteins to make up the new virion particles. Maturation is when the nucleic acid and the capsid proteins assemble, and then we get release, often by budding. This is uh, a mechanism used by those viruses that are enveloped, so they will then bud, or you get rupture of the cell, so the cell break open, and in this way the new virus particles or virions are then released, and they are ready to infect. In figure 13.14a, you can see the entry of viruses. In this case, a virus called the Toga virus, and this is by receptor-mediated endocytosis. You can see the virus attaches to the cell membrane through uh, specific proteins on the virus that recognizes receptor binding sites on the cell surface, and then the virus is engulfed. And you can see on the right hand side of this picture how this virus now moves into the cell through this process of endocytosis. This is an example of the entry of herpes virus by fusion. So um, you can see the virus bind to the cell surface to specific receptor uh, proteins on the cell surface, and then you get fusion of the viral envelope with the membrane surrounding the cell. So if that fusion takes place, then the virus is taken into the cell, and then the capsid is released into the cytoplasm, and the replication can commence. So again, the steps are attachment, penetration, uncoating, biosynthesis, maturation, and then release of the new virus particles. So release happens through the process of budding if the virus is an enveloped virus. This you can see in the figure. So there you have the viral capsid. This new virus particle then moves to the host cell plasma membrane. There are some viral proteins built into this host cell plasma membrane, and then you get budding. So the capsid um, comes together with the cell membrane into which viral proteins are built, and then you get budding, step 3, and step 4. You get release of this new virus particle or virion. So to show this process in a very nice electron micrograph, you can see in this case a lentivirus. So this is human immunodeficiency virus, and how this virus is released from the cell by budding. So you can quite clearly see how this happens. The capsid associates with the cell membrane. It then takes a bit of the cell membrane into which the viral proteins have already been built, and it then escapes um, and is ready to infect new cells. So table 13.3 compares bacteriophage and animal viral multiplication. So if we look at bacteriophages, remember the tail fibers attached to the cell wall protein. In animal viruses, attachment sites are plasma membrane proteins and glycoproteins, so receptor proteins. Then for bacteriophages, the viral DNA is injected. For animal virus, the capsid enters by receptor-mediated endocytosis or by fusion. Then to remove the capsid, in the case of animal viruses, this is done by enzymes, either supplied by the virus or supplied by the cell. This process is not required in the bacteriophages since the DNA, the naked DNA, has already been injected. So it's in the cytoplasm where the biosynthesis take place, but biosynthesis in animal virus can occur in the nucleus for DNA virus or the cytoplasm for most of the RNA virus. Then if we get a chronic infection step, this is called lysogenic infection in the case of bacteriophages, but in animal viruses this is known as latency or slow viral infections. It can also manifest in the form of a cancer. For release of the new virus particles, in the case of bacteriophages, the host cell is lysed, so it bursts open. It is then killed. In animal viruses, the enveloped viruses will release by budding, so they bud out and take with them a piece of the cell membrane of the cell. Um, and for viruses that do not have envelopes, so the non-enveloped viruses, they would rupture the plasma membrane, so break open the cell, and the cell will be killed. Let's look at the replication of a DNA containing animal virus. So start with step one at the top. We are looking here at a virus known as a Popova virus. So a Popova virus is a typical DNA containing virus that attacks animal cells. So we get attachment. So the virus attaches to specific proteins on the host cell. We then get entry into the cell and uncoating, so removal of the capsid. Then the DNA is released. So in step 3, a portion of the viral DNA is then transcribed. It produces messenger RNA that encodes early viral proteins and the biosynthesis commences. So in 4, the viral DNA is replicated and some viral proteins are made. And after the early steps, we then get what is known as late steps, so late translation, where the capsid proteins are synthesized when the virus is ready to form new virions. So there you can see the capsid proteins. These virions then mature in step 6, and in step 7, the newly formed virions. In figure 13.7, let's look at the pathways of multiplication used by various RNA-containing viruses. Now, for RNA-containing viruses, there are three possibilities. This will be A, it's single-stranded RNA of the plus or sense strand, such as you get for Picorna viridae, which includes, for example, poliovirus. Or B, where it's single-stranded RNA, but of the negative or antisense polarity. And this is what you get in the raptovirus. A good example is rabies virus. Or C, the genome will be double-stranded RNA. So it's plus and minus together in a double-stranded RNA. And this is what you find in the Rioviridae. A good example is the blue tongue virus. So let's start with one. We get attachment, you get entry, and we get uncoating. So in the case of A, where it's plus strand, you then get a negative strand made or transcribed from the plus viral genome, and this is then mRNA, so which will be transcribed from the negative strand. So once you've made negative strands, from that negative strands you will make more positive strands, which is the messenger RNA. And once the messenger RNA has been made, you can then get protein synthesis, and the capsid proteins are being made. The new virus particles are produced, so you get maturation and release. But in the case of B, the second column of that table, row of that table, when we have single-stranded RNA of the negative polarity, so then we must make messenger RNA or plus strand RNA, messenger, from the negative viral genome. And then viral proteins can be synthesized from this messenger RNA. So you then get the production of capsid proteins, packaging of the genetic material, maturation of particles, and release. And then in the third row, we have the example of double-stranded RNA, where messenger RNA is then produced actually inside the capsid of the protein, and the messenger RNA is then released into the cytoplasm. And once the messenger RNA is in the cytoplasm, ribosomes will recognize and start to produce proteins. So capsid proteins are then made. The viruses are formed, virus particles are formed, they mature, and you get release. And all of these newly released viruses are then ready to start a new infection cycle. So this is just a reproduction of the figure that we just did. So it's the same figure, but showing only row A for the single strand RNA, such as we get in Picorna viridae, of which a good example is the poliovirus. In this case, it's the same figure, but removing rows A and C, just looking at single-stranded negative polarity RNA, such as we find in the Rhabdoviridae. Good example is the rabies virus. And then again, the same figure, just showing row C. In this case, what happens if the genome is a double-stranded RNA, such as we find with viruses in the family Reoviridae. A good example is the blue tongue virus. So again, just the slide showing all three of these together, explaining the different pathways depending on the exact genome type. In figure 13.19, we look at the multiplication and inheritance processes of the Retroviridae. The Retroviridae include lentiviruses, species human immunodeficiency virus or HIV. Now, a retrovirus may become a provirus that replicates in a latent state, and it may then produce new retrovirus particles. So let's look at the steps. Step one, the retrovirus enters by fusion between the attachment spikes and the specific host cell receptors. Two, we then get uncoating and release of the two viral RNA strands of the HIV virus, and the viral enzymes reverse transcriptase, integrase, and protease are released. So this reverse transcriptase enzyme then copies the viral RNA to produce double-stranded DNA. This new viral DNA is then transported into the host cell's nucleus, where it is integrated into a host cell chromosome as a provirus by the enzyme viral integrase. The provirus may be replicated when the host cell replicates. So in the case of bacteriophages, remember we talked about a prophage in the lysogenic cycle. So here we talk about a provirus in this type of replication cycle. So in step 5, transcription of the provirus may also occur. This produces RNA for new retrovirus genomes and RNA that encodes the retrovirus capsid enzymes and envelope proteins. The viral proteins are then processed by this viral protease. Some of the viral proteins are moved and built into the host plasma membrane so that when budding takes place, such as you see in step 7, where mature retroviruses leaves the host cell, it would then acquire the envelope together with the attachment spikes, these proteins, viral proteins, already built into the membrane as it then buds out. And when the budding is complete, the virus is ready to start a new infection. In table 13.4, we look at a comparison between the biosynthesis of DNA and RNA animal viruses. So let's look at the first one. So we have DNA single-stranded, such as we find in the virus family Parvoviridae. And here we get the cellular enzyme that transcribes the viral DNA in the nucleus. So the cell actually provides the enzyme that will be used to transcribe the virus DNA. In the second row, we have double-stranded DNA, such as we find for herpes viridae, herpes simplex type 1, for example, or others, Papoviridae, or the pox viruses, or pox viridae. And here we get cellular enzyme that transcribes viral DNA in the nucleus, but in the case of the pox viruses, viral enzymes that transcribe viral DNA in the cytoplasm. In the third row, we have DNA viruses that utilize reverse transcriptase. That's the enzyme that converts RNA into DNA. And the virus family known for this is hepatnaviruses. That includes, for example, hepatitis B virus. So what happens here is that the cellular enzyme transcribes the viral DNA in the nucleus, and reverse transcriptase then copies messenger RNA to make new viral or DNA. So for the first of the RNA viruses, we use a positive strand as an example, so like we find in picornaviruses, such as poliovirus or rhinovirus. And the viral RNA functions as a template for synthesis of RNA polymerase, which then copies the negative strand RNA to make messenger RNA in the cytoplasm. In the second instance, we use RNA viruses with a negative strand genome, such as for rhabdoviruses, Rhabdoviridae, that includes rabies virus. So viral enzyme copies the viral RNA to make messenger RNA in the cytoplasm. So it's a virus enzyme. So the virus supplies the enzyme to copy its own RNA to make messenger RNA. This happens in the cytoplasm. For double-stranded RNA, Reoviridae, such as blue tongue virus, a viral enzyme copies the negative strand RNA to make messenger RNA in the virion, and this happens in the cytoplasm. And lastly, RNA viruses that utilizes reverse transcription. Best example, Retroviridae. It includes the lentiviruses, to which human immunodeficiency HIV virus belongs. Um, so the virus enzyme copies the viral RNA to make DNA in the cytoplasm, and then this DNA moves to the nucleus. Remember, where it's then incorporated into the chromosome as a provirus. In the case of viruses that cause cancer, we get activated oncogenes. These are genes coded for by the viruses, and they transform normal cells into cancerous cells. Transformed cells, these cancerous cells, have increased growth, loss of contact inhibition. They have tumor-specific transplant antigens and what is known as T antigens. The genetic material of oncogenic viruses becomes integrated into the host cell's DNA. Let's compare oncogenic viruses, those with DNA genomes and those with RNA genomes. So for the oncogenic DNA viruses, these are different families that are known to have uh, oncogenic potential. So the Adenoviridae, Herpes viridae, Pox viridae, Papoviridae, and Hepatna viridae. On the right, we have the oncogenic RNA viruses, only one family, and this is the family Retroviridae. So viral RNA is transcribed into DNA, which can integrate into the host DNA. And examples is the human T cell leukemia virus type 1 or HTLV type 2. In the case of latent and persistent viral infections, the virus would remain asymptomatic for a long period of time. So, a good example is cold sores, where you just occasionally get the cold sore, but the virus is always there, and just occasionally it breaks out of the asymptomatic stage and causes the sore. Shingles is another example. Can also be that the disease process occurs over a long period. This could generally be fatal, such as in the case of subacute sclerosing panencephalitis caused by measles. Figure 13.21 shows a graph that compares acute infection, latent and persistent viral infections. So acute infection is when the virus infects, produces new virus particles very fast, and the new virus particles are then released and continues with the infection of new cells. For a latent infection, the virus can remain in equilibrium with the host and not actually produce disease for long periods, often many years. A good example of a latent infection is the infection of the skin by herpes simplex virus, which produces cold sores. So this virus inhabits the host's nerve cells but causes no damage. It remains latent until it's activated by a stimulus factor such as fever or sunburn, and hence the term fever blister. And then you get where you see that green curve where the latent infection is activated. In contrast, persistent viral infections occur gradually over a long period of time. So they're not latent; they are chronic, um, but very gradually produce continually new uh, virus particles. And typically, such persistent viral infections can be fatal. So here we have the question, how is a persistent viral infection different from a latent viral infection? A, B, C, or D? And the answer is D, that the infectious virus gradually builds up over a long period of time. It is the latent infections that would do A, remain in equilibrium with the host, and B, can be reactivated, and C, appears very suddenly. So for a persistent infection, it is D, that the infectious virus gradually builds up over a long period of time. Now let's look at prions. These are very, very interesting pathogens. The name is derived from proteinaceous infectious particle. So these are proteins, infective proteins. They do not contain nucleic acid. They are protein only. And they can be inherited on the one hand, or they can be transmissible on the other hand by ingestion, by things like transplant procedure, or even from the surgical instruments using in operations. Some examples of spongiform encephalopathies is Crepey disease that you get in sheep, or Creutzfeldt-Jakob disease. It's a human disease. Same for Gerstmann-Straussler-Schenker syndrome, human disease. Fatal familial insomnia, human. And mad cow disease, which is the disease of cattle. And then Kuru, which is a prion disease of humans. For prions, the following terminology applies. So we get the prion protein C, and this is a normal cellular prion protein. It's on the cell surface, cells have it. We don't know exactly what that protein does, but it is a normal cellular protein. Then we get PRPSC. Now this SC stands for Scrapie or Scrapie. This was the first prion disease discovered, and therefore we use the SC from Scrapie. And this PRPSC, this is the disease protein, and it accumulates in brain cells where they form plaques or lesions in the brain. And this figure 13.22 shows how a protein can be infectious. So basically, if an abnormal prion protein, PRPSC, enters a cell, it changes the normal prion protein to PRPSC. It's almost like the bad apple in the box, which converts all the good apples into bad apples. So which can now change into another normal PRPC, resulting in an accumulation of the abnormal PRPSC. So if you look through the steps, so step 1, PRPC is a normal protein. It's produced by cells and it's secreted to the cell surface, where, as I say, we don't know exactly what the function is, but it is a necessary protein for the cell. And then 2, PRPSC, the disease prion, can now be acquired by ingestion, eating infected material, or it can be produced by a defective PRPC gene, in the case where the disease is inherited. So PRPSC then reacts with PRPC on the cell surface and changes it into PRPSC. So the new PRPSC now converts more PRPC. And then the new PRPSC is taken in possibly into the cell by receptor-mediated endocytosis, and then it accumulates in the endosomes. So PRPSC continues to accumulate as the endosome contents are transferred to lysosomes, and the result then is cell death. So prion disease occurs in three distinct mechanisms. One, the infectious prion disease. So in this case, the pathogenic form of prion protein is transmitted between animals or humans by, for example, ingestion of infected material. In the second instance, sporadic prion disease. This is a random misfolding of a normal healthy prion protein in an uninfected individual. So random happens by chance. But thirdly, the inherited prion disease. This is where we have a mutation in the normal prion gene that yields a protein that changes more often into the disease-causing form of prion. So just a brief comment on plant viruses and viroids. So how do plant viruses get into plants? Well, they typically enter through wounds, abrasions on the surfaces of the leaves, or very often via insects. So insects would feed on these leaves and then would transmit the virus from one plant to another in this manner. If we look at viroids, they are a specialized form of pathogen. These are infectious RNA molecules, so no capsid, like you have in the case of a virus. No capsid, just the RNA, which is infectious. And an example, and this occurs in plants. An example is the potato spindle tuber disease. Here we see an electron micrograph, a transmission electron micrograph, which shows linear on the left and circular, if you can see on the right, potato spindle tuber viroid.